Cell death is a fundamental biological process essential for life. Cell death is not simply a passive event, but a highly regulated process. It is controlled by specific molecular pathways that determine when and how cells die. But why do cells need to die? Let's visualize the transformation from a healthy cell to a dying cell. Elimination of damaged or potentially harmful cells, development of proper body structures, maintenance of cell population balance, homeostasis, protection against cancer and autoimmune diseases. In multicellular organisms, controlled cell death is crucial. The human body eliminates approximately 50 to 70 billion cells every day through regulated cell death. Without proper cell death mechanisms, multicellular life would be impossible. There are two main types of cell death we'll explore in this course. Apoptosis, or programmed cell death, is a controlled, orderly process. Necrosis, on the other hand, is accidental cell death, often resulting from acute injury or trauma. In summary, cell death is a regulated, essential process that maintains the balance between cell division and elimination. Disruptions in cell death mechanisms can lead to various diseases, including cancer and neurodegenerative disorders. Apoptosis is a highly regulated, energy-dependent process of programmed cell death. Apoptosis can be thought of as cellular suicide, a controlled dismantling of the cell. Unlike accidental cell death, apoptosis is highly regulated and energy-dependent. It involves controlled dismantling of cellular components and does not trigger inflammation. Apoptosis is crucial for normal development, tissue homeostasis, and immune system function. It helps eliminate cells that are damaged, infected, or no longer needed. The term apoptosis comes from Greek words meaning falling off, like leaves from a tree. Just as trees naturally shed their leaves, our bodies use apoptosis to remove unwanted cells. Understanding the controlled nature of apoptosis is key to comprehending how our bodies maintain cellular balance and health. Apoptotic cells display distinct morphological changes that occur in a sequential manner. Let's examine the morphological changes that occur during apoptosis, starting with a normal, healthy cell. The first visible change is cell shrinkage. The cell becomes smaller, cytoplasm becomes dense, and it loses contact with neighboring cells. Next, chromatin condensation occurs. The nuclear material clumps together, and the DNA begins to fragment. This is followed by nuclear fragmentation, or karyorrhexis, where the nucleus breaks into fragments and the nuclear envelope disintegrates. Membrane blebbing follows, where the cell membrane forms bubble-like protrusions due to the breakdown of the cytoskeleton. Finally, the cell fragments into membrane-bound apoptotic bodies containing cellular components and nuclear fragments. These bodies are then recognized and removed by phagocytes. These morphological changes occur in a sequential manner and are hallmarks used to identify apoptotic cells under a microscope. These distinct morphological features are used as diagnostic criteria in pathology and are fundamentally different from necrotic features like cell swelling and membrane rupture. The extrinsic pathway of apoptosis is triggered by external death signals and occurs at the cell membrane. First, specialized death receptors are found embedded in the cell membrane. These include Fas, TNF receptor, and TRAIL receptors. Death ligands such as FASL, TNF-alpha, and TRAIL approach the cell and bind to their specific death receptors. When death ligands bind, the receptors cluster and recruit adapter proteins like FADD and procaspase 8, forming the death-inducing signaling complex, or DISC. Within the DISC complex, procaspase 8 becomes activated through a process called proximity-induced dimerization and autoprocessing. Active caspase 8 then cleaves and activates executioner caspases such as caspase 3, caspase 6, and caspase 7. These executioner caspases dismantle the cell by cleaving structural proteins, fragmenting DNA, and preparing the cell for disposal. This completes the extrinsic pathway of apoptosis, where external death signals trigger a cascade of caspase activations leading to programmed cell death. The intrinsic pathway of apoptosis is activated by cellular stress signals that originate from within the cell. Various types of cellular stress can trigger this pathway, including DNA damage, oxidative stress, and growth factor withdrawal. When cellular stress is detected, the intrinsic pathway centers on the mitochondria. These organelles contain cytochrome C, a critical protein for apoptosis. In response to stress signals, pro-apoptotic proteins in the BCL2 family permeabilize the outer mitochondrial membrane, leading to the release of cytochrome C into the cytosol. Once in the cytosol, cytochrome C binds to a protein called APAF1. These proteins, along with ATP, form a wheel-like structure called the apoptosome. This complex recruits and activates pro-caspase 9. Once activated, caspase 9 cleaves and activates executioner caspases, including caspase 3, caspase 6, and caspase 7. These executioner caspases then cleave various cellular substrates, leading to the biochemical and morphological changes characteristic of apoptosis. To summarize, the intrinsic pathway of apoptosis begins with cellular stress, causes mitochondrial damage, leads to cytochrome C release and apoptosome formation, activates caspase 9, which then activates executioner caspases, ultimately resulting in controlled cell death. The BCL2 family of proteins plays a crucial role in regulating the intrinsic pathway of apoptosis. These proteins are categorized into two main groups based on their function in apoptosis. Pro-apoptotic proteins, such as BAX, BAD, BID, and BIM, promote cell death by facilitating mitochondrial permeabilization. Anti-apoptotic proteins, including 2-TLXL, MKL1, BCLW, and A1, prevent apoptosis by inhibiting the action of pro-apoptotic proteins. All BCL2 family proteins share structural similarities, containing one or more BCL2 homology domains, known as BH domains. These domains, labeled BH1 through BH4, determine the function of each protein. The BH3 domain is particularly important for pro-apoptotic activity. Let's examine how these proteins interact at the mitochondrial membrane. Pro-apoptotic proteins like BAX can insert into the outer mitochondrial membrane. Anti-apoptotic proteins like BCL2 can bind to and neutralize pro-apoptotic proteins, preventing their activity. The balance between pro-apoptotic and anti-apoptotic BCL2 proteins determines whether a cell undergoes apoptosis. When pro-apoptotic proteins dominate, they overcome the protective effect of anti-apoptotic proteins, leading to cell death. Conversely, when anti-apoptotic proteins predominate, they prevent activation of the death pathway, promoting cell survival. When pro-apoptotic proteins prevail, they induce mitochondrial outer membrane permeabilization, or MOEMP. Activated BAX and BAC oligomerize and insert into the mitochondrial outer membrane. This creates pores in the membrane, allowing cytochrome C and other pro-apoptotic factors to leak into the cytosol. The release of cytochrome C triggers the formation of the apoptosome, activating caspases and executing cell death. In summary, the balance of BCL2 family proteins regulates MOEM, which represents the point of no return in the intrinsic apoptotic pathway. The interplay between these proteins is tightly regulated and represents a critical control point in determining whether a cell lives or dies. After a cell undergoes apoptosis, its remnants must be efficiently cleared from the body. Apoptotic cells display "eat me" signals on their surface. The most important of these is phosphatidylserine, a phospholipid that flips from the inner to the outer leaflet of the plasma membrane. This phospholipid normally resides in the inner leaflet of the membrane, but during apoptosis, it flips to the outer leaflet, serving as a signal for phagocytes. Phagocytes, such as macrophages, recognize these "eat me" signals through specific receptors on their surface. These receptors bind to phosphatidylserine and other signals, initiating the engulfment process. The process of phagocytic clearance of apoptotic cells is called efferocytosis. It involves engulfment and digestion of the cellular debris. The phagocyte extends its membrane around the apoptotic cell, completely engulfing it. Once inside, the apoptotic cell contents are broken down by enzymes in the phagocyte, completing the removal process. Timely clearance of apoptotic cells is crucial. It prevents the apoptotic cells from progressing to secondary necrosis. When clearance is efficient, tissue remains healthy with no inflammation. However, if clearance fails, secondary necrosis occurs, leading to the release of cellular contents that trigger inflammation and tissue damage. To summarize, apoptotic cell clearance is a crucial process that maintains tissue health. Apoptotic cells expose eat me signals. Phagocytes recognize and engulf these cells through efferocytosis, and this process prevents inflammation by avoiding secondary necrosis. Necrotic cells display distinct morphological changes that differentiate them from other forms of cell death, like apoptosis. Let's start by examining a healthy cell with normal morphology before exploring the changes seen in necrosis. The first defining feature of necrosis is cell swelling, also known as oncosis, which contrasts with the cell shrinkage seen in apoptosis. Let's examine the 5 key morphological features of necrosis. First, cellular swelling occurs as the cell loses its ability to maintain ion homeostasis. Second, the plasma membrane ruptures, losing its integrity and allowing cellular contents to leak into the surrounding tissue. Third, organelles such as mitochondria and the endoplasmic reticulum swell and lose their function. This is especially visible in mitochondria, which can rupture. Fourth, DNA undergoes random degradation in necrosis, unlike the organized fragmentation seen in apoptosis. Finally, the cell releases its contents, including enzymes and damage-associated molecular patterns, into the extracellular space. This spillage triggers inflammation in surrounding tissues. Let's compare these necrotic features with apoptosis, which shows very different characteristics. While necrosis involves swelling and rupture, apoptosis is characterized by cell shrinkage and membrane blebbing. These distinct morphological features help pathologists and researchers identify and distinguish necrotic cell death from other forms of cell death in tissues. Necrosis involves several distinctive biochemical changes that differ from apoptosis. While a healthy cell maintains biochemical balance, necrotic cells experience rapid and chaotic biochemical deterioration. The first major biochemical feature is rapid ATP depletion. As cellular energy production fails, metabolic processes collapse. Ion homeostasis disruption occurs as membrane pumps fail, unable to maintain proper ion gradients across the cell membrane. This leads to massive calcium influx into the cell, which activates various degradative enzymes. Proteases such as calpains become activated, resulting in widespread protein degradation throughout the cell. Reactive oxygen species production increases dramatically, causing oxidative damage to cellular components. Finally, random DNA degradation occurs due to nonspecific nucleases, creating irregular DNA fragments, unlike the ordered fragmentation seen in apoptosis. These biochemical features collectively characterize necrosis as a chaotic cellular breakdown process. Necroptosis is a regulated form of necrotic cell death that combines features of both apoptosis and necrosis. Unlike apoptosis and necrosis, necroptosis shares characteristics with both processes. It is programmed like apoptosis, but exhibits morphological features of necrosis. The key distinguishing features of necroptosis include programmed regulation like apoptosis, but cell swelling and inflammatory response like necrosis. The main molecular players in necroptosis are receptor interacting protein kinase 1, REP3, and mixed lineage kinase domain-like protein, LKL. In this pathway, RIC1 phosphorylates at 3, which then phosphorylates MLKL. Once activated, MLKL forms pores in the plasma membrane, leading to cell swelling and rupture. Necroptosis is typically activated when apoptosis is inhibited, such as during viral infections that block caspases, yet death receptor signaling is still active. This unique activation mechanism, apoptosis inhibition plus death receptor activation, creates a backup cell death pathway when apoptosis fails. Understanding necroptosis is crucial because it represents an important fail-safe mechanism for eliminating cells when apoptosis is compromised, particularly during viral infections. This section explores the molecular mechanisms of necroptosis, a form of programmed necrotic cell death. Necroptosis begins with activation of death receptors such as TNFR1. Normally, these receptors would trigger apoptosis, but when caspase 8 is inhibited, the cell is directed toward necroptosis instead. Caspase-8 inhibition is a critical trigger for necroptosis. When this enzyme is blocked, it cannot cleave RIPK1 and RIPK3, allowing these proteins to interact and form a complex. In the absence of caspase-8 activity, RIP1 and RIC3 interact to form a complex known as the necrosome. This complex is the central signaling hub that drives necroptosis forward. Within the necrosome, RIP3 phosphorylates MLKL, the mixed lineage kinase domain-like protein. This phosphorylation is a critical step that activates MLKL and transforms it into the executioner of necroptosis. Once phosphorylated, MLKL undergoes a conformational change that allows it to form oligomers. These oligomers are the active form of MLKL that can translocate to the plasma membrane. At the plasma membrane, MLKL oligomers form pores or otherwise disrupt membrane integrity. This leads to rapid influx of ions, cell swelling, and ultimately membrane rupture with the release of cellular contents. To summarize the molecular mechanisms of necroptosis, death receptor activation combined with caspase-8 inhibition leads to necrosome formation. RIP3 then phosphorylates MLKL, which oligomerizes and translocates to the plasma membrane. There, it causes membrane permeabilization, resulting in cell death with necrotic features. Pyroptosis is a form of inflammatory cell death that is distinct from apoptosis and necrosis. Unlike apoptosis, which is non-inflammatory, pyroptosis triggers a strong inflammatory response. Pyroptosis is triggered by inflammasome activation in response to pathogen signals or cellular stress. The inflammasome is a multi-protein complex that assembles when activated, bringing together NLRP3, ASC, and procaspase-1. This assembly activates caspase-1, which is the key enzyme in pyroptosis. Active caspase-1 cleaves gasdermin D, a cytosolic protein with N and C terminal domains. Cleavage separates these domains, releasing the N-terminal fragment that forms pores in the cell membrane. These N-terminal fragments oligomerize to form pores in the plasma membrane, allowing water influx and cell swelling. The formation of these pores leads to water influx, causing the cell to swell. Pyroptosis is characterized by the release of pro-inflammatory cytokines, particularly IL-1 beta and IL-18. These cytokines exit through the gasdermin D pores and trigger inflammation in surrounding tissues. Eventually, the cell membrane ruptures completely, releasing all cellular contents and amplifying the inflammatory response. This inflammatory cell death helps eliminate pathogens, but can also contribute to tissue damage in inflammatory diseases. To summarize, pyroptosis is a distinct inflammatory form of cell death triggered by inflammasome activation, leading to caspase-1 activation, gasdermin D pore formation, and the release of pro-inflammatory cytokines. Autophagy is a critical cellular process where cells digest and recycle their own components. The term autophagy comes from the Greek words auto, meaning self, and phagi, meaning eating. While autophagy primarily serves as a survival mechanism, helping cells maintain homeostasis, excessive autophagy can paradoxically lead to cell death. To understand autophagy, let's observe the process in a healthy cell. The process begins with the formation of a membrane structure called a phagophore. The phagophore expands to form a double membrane structure called an autophagosome, which engulfs cellular components to be degraded. Next, the autophagosome fuses with a lysosome, which contains digestive enzymes. The resulting structure, called an autolysosome, breaks down the engulfed components and recycles them for cellular use. While controlled autophagy is beneficial, excessive autophagy can lead to a type of programmed cell death known as autophagic cell death. This occurs when the cell forms too many autophagosomes, excessively degrading essential components. Eventually, this leads to cellular collapse and death. Autophagic cell death has both similarities and differences when compared to apoptosis and necrosis. Unlike necrosis, both autophagy and apoptosis maintain membrane integrity and cause minimal inflammation. However, autophagy's primary purpose is cellular recycling and survival, while apoptosis is dedicated to programmed cell elimination. To summarize the key points about autophagy, it's primarily a pro-survival mechanism that can paradoxically lead to cell death when excessive. It shows significant crosstalk with apoptosis pathways and is critical for cellular homeostasis. Dysregulation of autophagy has been linked to various diseases, including cancer and neurodegenerative disorders. Cell death, particularly apoptosis, plays a crucial role in normal development, sculpting tissues and removing unnecessary cells. During development, specific cells are programmed to die at precise times and locations, allowing for proper organ formation. Digit formation provides a classic example of developmental apoptosis. In early stages, embryonic hands have webbing between the fingers. Programmed cell death eliminates this interdigital tissue, creating separated fingers. Without proper apoptosis, humans would have webbed digits. In nervous system development, the brain initially produces an excess of neurons. Through a process called neural pruning, neurons that fail to make proper connections undergo apoptosis. This refines neural circuits and improves efficiency. Immune system development relies on apoptosis to eliminate potentially harmful self-reactive lymphocytes. In the thymus, T cells undergo selection. Those that recognize self-antigens too strongly are eliminated through apoptosis. This process, called negative selection, prevents autoimmune diseases by removing cells that would attack the body's own tissues. In reproductive system development, all embryos initially have both male and female reproductive duct systems. In male development, apoptosis eliminates the Müllerian ducts, while hormones preserve the Wolffian ducts. Conversely, in female development, the Wolffian ducts degenerate through apoptosis, while the Müllerian ducts develop into the fallopian tubes, uterus, and upper vagina. Cell death plays a crucial role in maintaining tissue homeostasis in adult organisms. Tissue homeostasis is the delicate balance between cell proliferation and cell death. This balance ensures that tissues maintain their proper size and function throughout an organism's life. The intestinal epithelium demonstrates this balance clearly. New cells are constantly generated in the intestinal crypts. These cells migrate up the villus and eventually undergo apoptosis at the tip, being shed into the intestinal lumen. In the skin, basal cells constantly divide and migrate upward. As they move toward the surface, they undergo terminal differentiation. Eventually, these cells die and are shed from the skin surface as new cells replace them from below. Red blood cells provide another example of tissue homeostasis. Young red blood cells have a lifespan of approximately 120 days in circulation. As they age, they undergo various biochemical changes. Finally, senescent red blood cells are recognized and removed by macrophages in the spleen and liver. The body produces approximately 200 billion new red blood cells daily, with an equal number being removed by macrophages. When the balance between cell proliferation and death is disrupted, tissue homeostasis breaks down. Excessive proliferation with insufficient cell death can lead to cancer, tumors, or hyperplasia. Conversely, excessive cell death without adequate replacement leads to tissue atrophy and degeneration. These imbalances are at the root of many diseases, including degenerative conditions, cancer, autoimmune disorders, and aging-related pathologies. Cell death dysregulation contributes to various diseases. The balance of cell death is critical for health. Dysregulation can occur in two ways. Excessive cell death leads to tissue destruction and organ failure. Insufficient cell death allows abnormal cells to persist and accumulate. Excessive cell death contributes to several categories of diseases. Neurodegenerative diseases like Alzheimer's and Parkinson's involve inappropriate death of neurons. Ischemic injuries such as strokes and heart attacks cause rapid cell death due to oxygen deprivation. Some autoimmune disorders involve excessive destruction of specific cell types. Insufficient cell death also contributes to several disease categories. Cancer develops when damaged or mutated cells fail to undergo cell death, allowing tumors to form and spread. Some autoimmune disorders occur when self-reactive immune cells fail to be eliminated. Persistent infections can develop when infected cells evade cell death mechanisms. Dysregulation of cell death pathways can occur at the molecular level. Alterations in apoptosis, necrosis, and autophagy pathways can all contribute to disease development. These pathways interact in complex ways, and their dysregulation can lead to various disease states. Understanding these dysregulated pathways provides opportunities for developing targeted therapies. By targeting specific cell death mechanisms, we can develop more effective treatments for these diseases. Cancer cells differ from normal cells in their ability to evade cell death mechanisms, which is a hallmark of cancer. One of the primary ways cancer cells evade apoptosis is through overexpression of anti-apoptotic proteins such as BCL2 and IAPs. Cancer cells also downregulate pro-apoptotic proteins like BAX and BAC, further protecting themselves from cell death signals. Another crucial mechanism is mutation of the p53 tumor suppressor gene, which normally initiates apoptosis in response to DNA damage. Cancer cells can also alter death receptor signaling, preventing external death signals from activating the extrinsic apoptotic pathway. Understanding these mechanisms has led to the development of various cancer therapies targeting apoptosis pathways. These include BH3 mimetics that target BCL2 family proteins, IAP antagonists, strategies to reactivate p53, and death receptor agonists. For example, BH3 mimetics are small molecules that mimic BH3-only proteins, binding to anti-apoptotic BCL2 family proteins and preventing them from inhibiting apoptosis. By understanding the mechanisms through which cancer cells evade apoptosis, researchers have developed targeted therapies that aim to restore normal cell death pathways, offering new approaches to cancer treatment. Cell death pathways are crucial targets for therapeutic intervention in various diseases. There are three major approaches to therapeutically targeting cell death pathways. Pro-apoptotic drugs activate cell death in cancer cells. These include BH3 mimetics, IAP antagonists, and death receptor agonists. Anti-apoptotic drugs prevent excessive cell death in degenerative diseases, including caspase inhibitors, antioxidants, and BCL2 family stabilizers. Necroptosis and pyroptosis modulators control inflammatory cell death through RIPK inhibitors, MLKL inhibitors, and inflammasome inhibitors. Let's examine venetoclax, a BCL2 inhibitor, as a case study in therapeutic targeting of cell death. Venetoclax specifically targets BCL2 proteins which are overexpressed in certain leukemias like chronic lymphocytic leukemia and acute myeloid leukemia. By binding to BCL2, venetoclax prevents it from inhibiting apoptosis, thus restoring the cancer cell's ability to undergo programmed cell death. Despite successes like venetoclax, therapeutic targeting of cell death faces several challenges. Future directions include combination therapies, biomarker-guided treatment approaches, novel delivery systems, and integration with immunotherapies. Therapeutic targeting of cell death pathways represents a promising frontier in disease treatment, particularly for cancer and degenerative disorders. Detecting cell death requires specialized techniques that can identify specific characteristics of dying cells. Several methods are commonly used in research and clinical settings to detect and distinguish between different types of cell death. The TUNEL assay, or terminal deoxynucleotidyl transferase (DUTP-NICN) labeling, detects DNA fragmentation during apoptosis. This technique uses the enzyme terminal deoxynucleotidyl transferase to add labeled nucleotides to the 3' hydroxyl ends of fragmented DNA. The TUNEL assay offers several advantages and has some limitations. Annexin 5 staining detects phosphatidylserine exposure on the cell surface, which is an early event in apoptosis. During early apoptosis, phosphatidylserine flips from the inner to the outer leaflet of the plasma membrane, where annexin 5 can bind to it. When combined with a viability dye like propidium iodide, this method can distinguish between early apoptosis, late apoptosis, and necrosis. Caspase activity assays measure the enzymatic activity of caspases, which are key executioners of apoptosis. These assays use fluorogenic or chromogenic substrates that produce a measurable signal when cleaved by active caspases. Caspase assays are particularly useful for quantitative measurements, but have specific limitations. Mitochondrial membrane potential measurement detects the loss of potential across the inner mitochondrial membrane, which is a key event in intrinsic apoptosis. Fluorescent dyes like JC1 or TMRM accumulate in functional mitochondria, but are released when the membrane potential collapses during apoptosis. This method is useful for detecting early apoptotic events, but has specific limitations to consider. Electron microscopy provides the highest resolution visualization of the ultrastructural changes that occur during cell death. It can reveal detailed morphological features like chromatin condensation, membrane blebbing, and organelle disruption that distinguish between apoptosis and necrosis. Understanding cell death is fundamental to modern biology and medicine. Cell death is not simply the end of cellular life, but a complex process with diverse forms and functions. Apoptosis is a controlled, programmed form of cell death that causes minimal inflammation and allows for proper cellular recycling. In contrast, necrosis is typically uncontrolled and leads to cell rupture, triggering inflammatory responses. The key differences between these cell death mechanisms have profound implications. Understanding cell death impacts our knowledge of development, disease mechanisms, and potential therapeutic interventions. Future research will continue to uncover new cell death mechanisms, develop targeted therapies, and improve our understanding of how cells make life or death decisions. Cell death research continues to reveal the delicate balance between life and death at the cellular level, offering new opportunities for medical advancement.